Hello dear students, welcome back to my channel. In this video, we are going to learn about a wonderful topic named transcription. Transcription of DNA which takes place in our cell. So in this video, I will detailedly explain you about the transcription of DNA. In general, transcription means a process of converting an audio or video into an written text. That is, the information will be in an audio format and it will be transcribed into the text. This is the general definition we give for transcription. In biology, we say that transcription is the copying of DNA sequence into a messenger RNA, which is essential for the synthesis of proteins. In our video, I have detailedly explained about the DNA structure and its function. You can go through that video to learn more about DNA. As we all know that DNA is a double helical structure having the nucleotides thymine, cytosine, adenine and guanine. But RNA is a single-stranded one with nucleic acids, uracil, cytosine, adenine and guanine. In both of this, you can see the differentiation that here in DNA we have thymine as one nucleotide, but here in RNA we have uracil as the nucleotide. So thymine is replaced with uracil. And another important difference is DNA is a double-stranded one, but RNA is a single-stranded nucleic acid. So transcription of DNA into RNA is nothing but the copying of the information that the DNA contains in its sequence. It is copied as an mRNA, and from the mRNA it is translated into protein, a functional one. So our body requires protein for the proper function. For the production of the protein, the information is stored in the DNA. So from the DNA, it is copied into an intermediate structure that is called mRNA, and from mRNA, it produces protein. Why our body needs protein? What is the importance of this proteins? So proteins are the macronutrients. It is made up of amino acids. So it acts as the primary building blocks for the body cells, tissues, organs and muscles and bones. Protein is vital for maintaining good health as well as supporting our various bodily functions. What are the functions? Let us see some examples. So the protein is important for our growth and maintenance of tissues, to repair our tissues throughout the body including muscles, skin, hair and nails. And in enzyme production. So the proteins are enzymes. So it takes place in the biochemical reactions in and out of the cells for the energy production. Mainly it takes place in digestion of our body. The proteins are required. The proteins are nothing but the enzymes. All enzymes are proteins, but all proteins are not enzymes. And then hormone regulation. Some proteins, they act as the chemical messengers. So it helps in the communication between cells, tissues and organs. Example we can have here is insulin, which regulates our body sugar. Immune system, here some proteins act as antibodies that fight infection and protect the body from harmful invaders like bacteria and viruses. And next comes the structural support, and here some fibrous proteins like keratin, collagen, they helps in the rigidity and flexibility of cells and tissues, forming the structure of bones, ligaments and skin. And coming to fluid and pH balance, the proteins such as albumin and globulin, it helps in maintaining fluid balance in the blood. And they also act as buffers to regulate the acid-base balance in the blood and other body fluids. And last one is transport and storage. Here also some nutrients like vitamins, minerals and oxygen is transported throughout the bloodstream to the various cells through blood. That is the protein present in blood is the hemoglobin. So the proteins are very essential for maintaining a good health and to support our bodily functions. So the informations for a particular protein to be produced in our cell is stored in DNA. And from the DNA it is copied into mRNA, and from mRNA we get the proteins. So looking into the structure of the protein, we have different structures for the protein depending upon their functions. So amino acids are the building blocks of the proteins. These are the smaller units of proteins. So these amino acids, they combine together. See, the first one we call it as a primary structure. Okay, phosphodiester bond, polypeptide chain. And then it also forms a secondary structure. Here you can see as alpha helix or as a sheet. It forms this structure according to its function. And it also has the tertiary structure. And last one is the quaternary structure. This structure is familiar in our blood cell, that is heme. Hemoglobin, we have a protein in our blood, no? That structure with the quaternary structure of a protein. So the structure of protein also differs according to its function. We have two different types of cells, prokaryotic cells and eukaryotic cells. So the process of transcription is same. The DNA is copied into mRNA and mRNA into protein. So the process is same, but the location is different. In prokaryotic cells, in the cytoplasm, the transcription takes place because prokaryotic cell does not have a well-defined nuclear membrane. So this process takes place in the cytoplasm. But in eukaryotic cells, we have a well-defined nuclear membrane. Inside we have the nucleus. So the transcription takes place inside the nucleus. And the processing of RNA also takes place inside the nucleus, and then it is transported outside the nucleus in the cytoplasm where the protein synthesis takes place. So the conversion of mRNA into this one protein, we call it as translation. There are three steps for transcription: initiation, elongation and termination. Let us see one by one. Initiation. So the process to be initiated, there should be some starter sequence so that the enzymes can recognize that sequence and it starts its transcription. So that start sequence we call as the promotase. Okay, it serves as a recognition and binding site for RNA polymerase. So the enzymes which helpful for the conversion of DNA into RNA, we call it as RNA polymerase. And we have two different types of cells, that are prokaryotic cells and eukaryotic cells. So the promoters are also different in it. In prokaryotic cells, we have TAT or Prenov box, we call this a promoter region. So this is the nucleotides. T stands for thymine and A stands for adenine. If you see this sequence, the enzyme can recognize that. After that, the DNA should be transcribed into mRNA. So the RNA polymerase attaches in this, after this, the region, it attaches and it continues the transcription process. And eukaryotic, the starter sequence is TATA box, Tata box. So here the RNA polymerase, it recognizes, if it recognizes this sequence, then once it is getting ready for the transcription of DNA into mRNA. And the function of RNA polymerase is catalyzing the synthesis of RNA from DNA template. So the first picture shows the promoter region in prokaryotic cells. Okay. So along with the sigma factor, the RNA polymerase, it attaches with the DNA by recognizing the promoter sequence TAT box. Okay. As well as in the eukaryotic region also, it recognizes the sequence TATA box. It recognizes this region and after that transcription takes place. So its region is recognized with the help of RNA polymerase enzyme. The next step is elongation. So once the RNA polymerase is attached with the DNA strand, it forms a transcription bubble. And the transcription takes place in a 5' to 3' direction. Here in DNA we have two strands. No, it's a double helical strand. But the RNA transcription takes place in any one of the strand. We call it as the template strand. And the difference is here you can see adenine always pairs with thymine in the DNA. But once it is transcribed, when it forms the RNA, the thymine is replaced with the U, that is the uracil. So the adenine, it combines with uracil. See, adenine, it combines with uracil. And guanine, it combines with cytosine. And thymine in the DNA, it combines with adenine. Okay. But when adenine is in DNA, then uracil will be attached in the RNA. That is the difference. And like this, the addition of sequence takes place and the mRNA is formed in the DNA strand. Next one is termination. So here also it takes place in eukaryotic cell and this takes place in the prokaryotic cells. Termination. So in eukaryotic cell, the termination we call as intrinsic termination. Here they have the stop sequence. If it recognize the continuation of the nucleotides A, G, C, GC rich region, it gets stopped. You can see here, and the polyadenine will be attached at the end of the 3' end of mRNA. As well as here we call it as row dependent termination. That means the row factor will attach with the RNA polymerase and it gives signals to stop the transcription. Here also we have the hairpin like structure. You can see it is rich in guanine and cytosine. Once the pre-RNA is formed from the DNA, it has exon and intron. What do you mean by that? Exon means it is the coding region. Intron means it is non-coding region. So we need only this exon. Intron has to be removed because it is of no use. There is nothing, information is present there. So the information for the protein formation is present only in the exon. So the introns are spliced and the exons are joined together to form a mature mRNA. So this process we call as splicing. And once the mature mRNA is formed, it is attached with G cap in the 5' end and polyadenation in the 3' end. This is the precaution for the mRNA to be degraded by the other enzymes. So when it is attached like this in the 5' end and 3' end, it is safeguarded from the degradation by other enzymes. Now let us learn about the RNA splicing. How does this takes place here and how the addition of tail and a G cap forms, you can see in the slide. RNA splicing. What do you mean by that? RNA splicing, that means cutting of the non-coding regions, that is introns. It takes place with the help of the enzyme spliceosomes along with some small ribonuclear particles, that is named as U1, U2, U3, U4, U5 and U6. This flowchart will tell you about the splicing of the introns. So now see, this is the premature mRNA having both intron and exons. Now this intron has to be removed. So here the starting region you have the GC, guanine and uracil rich region, and the end we have the AG. So these are the recognition sites for the spliceosome. So once it is recognized, first small nuclear protein binds to the premature RNA U1 at the first site, and then U2 it attached with the other end, and next it is followed by U4 and U6, and finally U5 get attached with the thing. Now you can see a small ring like structure formation, you can see here. So at first what happens, this complex only we call it as spliceosomes. Okay, at first what happens, at the 5' end it gets spliced and it forms a lariat structure. The structure we call as lariat structure. And next what happens, RNA is cleaved at the 3' end and now it gets completely detached from the exons and the two exons are joined together, and the introns which is removed, that is lariat structure, or this structure we call it as lariat structure, and this is degraded. Once the introns are excised from the exons, it will be degraded by the enzymes. There is another method of RNA splicing, that is alternate splicing. Here you can also see that the introns, the green structure, it will be removed and the exons will be joined together. The formation will be different. See, exon 1, 2, 3, it combines to form protein A, but if 1, 2 and 4 combines, it gives another protein B. So there will be the structural difference as well as also the functional difference between them. So this process we call as the alternate splicing. Summary, we can see that in a eukaryotic cell, that is human, we come under eukaryotic type of cells. Here the transcription of DNA into mRNA takes place inside the nucleus, and once the RNA is formed and it becomes a mature mRNA, it gets transported into the cytoplasm, and in cytoplasm the other process called translation takes place. That is nothing but the information in the RNA will be converted into proteins. So in our upcoming slides I will discuss with you in detail about translation, what do you mean by that, and how the proteins are formed. Stay with us to get more information about biology. Thank you for listening and happy learning.